Welcome back to the deep dive. We're plunging into some fascinating biological currents today. It all kind of kicks off with that old saying, mad as a March hare.
Yeah, that phrase, it's been around for centuries, hasn't it? And it perfectly captures uh, some really visible animal behavior linked to reproduction.
Exactly.
So for this deep dive, we're drawing heavily from a chapter in Animal Physiology
Yeah.
by Hill, Wise, and Anderson.
A classic text.
Absolutely. Our goal here is to really unpack the physiological concepts, the mechanisms, the different strategies animals use to reproduce. We'll look at comparisons, why certain strategies evolve their adaptive significance, and even some of the cool experimental methods scientists use to figure this stuff out.
So, kind of a shortcut through the textbook chapter, but making it really come alive with examples.
That's the idea. Think of it as your guided tour through the complex dance of life.
Okay, because what looks like chaos, like those hares, it's often anything but, right?
Oh.
It's intricate signaling, hidden hormones. Get ready for some surprises. Synchronized mating, weird parenting, all driven by invisible biology. Okay, let's unpack this. So those March hares, Lepus europaeus, they're famous for that boxing and dashing around in spring.
Yeah. Very visible.
But the surprising thing is it's not always males fighting males. Often it's a female literally fighting off a male because she's just not receptive yet.
And that simple observation, that boxing match, it tells us two really key things right away.
Okay.
First, reproduction is often seasonal. It doesn't just happen anytime. And second, female receptivity is specific. There's a window. Outside that window, it's a definite no.
Which leads us to a really core difference in how mammals handle ovulation, right?
Induced versus spontaneous.
Exactly. That's a fundamental split. So induced ovulator, like our hares and rabbits, also some cats, shrews, camelids, they only ovulate, meaning release eggs, after the physical act of mating.
So copulation is the trigger. It is. It's like an on-demand system, and it's incredibly efficient. Rabbits, for example, have almost 100% fertilization success because the eggs only appear when sperm are definitely around.
Wow, nearly 100%. Okay, so that's induced. What about spontaneous ovulators?
That's the other path. Most mammals, actually humans, dogs, cows, sheep, most rodents are spontaneous ovulators. Their bodies release eggs based on an internal hormonal cycle, pretty much regardless of whether mating happens.
So it's pre-scheduled, in a way.
You could say that. And it's interesting because even though induced ovulation seems like it would guarantee better fertilization, it's only found in a minority of mammals. Physiologists spend a lot of time studying the intricate mechanisms behind both.
Okay, so how does that triggering mechanism work in an induced ovulator like the rabbit? How does mating flip that switch?
It's a fantastic example of what we call a neuroendocrine reflex. It involves both nerves and hormones.
Okay.
So copulation physically stimulates nerves, sensory nerves, in the female cervix and other areas.
Right.
Those nerve signals travel up to the brain. This stimulates specific neurons in the brainstem, which then increase the release of norepinephrine, a neurotransmitter, right into the hypothalamus.
Okay, step by step.
That norepinephrine then tells specialized neurosecretory cells in the hypothalamus to release a big pulse of gonadotropin-releasing hormone, GnRH.
GnRH, okay.
This GnRH travels through a special little blood vessel system, the portal system, directly to the anterior pituitary gland.
Got it.
And the GnRH hits the pituitary and signals it to release a massive surge of luteinizing hormone, LH, into the general circulation.
The LH surge.
That's the key. This huge LH wave peaks just an hour or two after mating, and it's the LH that directly tells the ovaries, release the eggs.
Wow, that's incredibly fast and precise, like a biological Rube Goldberg machine almost.
It really is a perfectly coordinated cascade. What's fascinating here is how precisely this whole system is coordinated, almost like a biological cascade.
But this single event, this mating and ovulation, it fits into a bigger picture, right?
Right.
An annual rhythm.
Absolutely. Wild rabbits, for instance, are seasonal breeders. They're generally only reproductively active during the warmer months, typically spring and summer.
And the males get ready too.
They do. As the days get longer after the winter solstice, male rabbits experience testicular recrudescence. Basically, their tests, which might shrink in winter, rapidly regrow and ramp up sperm production.
And the females?
Females enter estrus, or heat, which is that state of sexual receptivity. This state is brought on by rising estrogen levels from their developing ovarian follicles. It's beautifully synchronized, so females are receptive only when they actually have mature eggs ready.
Makes sense. The environment must play a huge role in all this timing.
Oh, dominant. Absolutely dominant. Reproductive success hinges on environmental conditions. We're talking physical factors like temperature, but also biological ones.
Nice.
Food availability is huge.
Right.
Predator pressure, even the presence of others of their own species. Think about fish larvae. There are documented cases where entire cohorts starved because their hatching wasn't synchronized with the plankton bloom they needed for food.
So alignment is critical. What cues do animals use to get this timing right? Temperature seems obvious, maybe?
Temperature is a cue, often linked to food, but it can be unreliable. Weird warm spells in winter, cold snaps in spring.
Yeah, happens all the time.
But photoperiod, the length of daylight, that's different. It's mathematically precise, completely unambiguous day after day, year after year. It's a very reliable clock.
So how do animals actually use day length? Is it just one mechanism?
It's actually quite diverse. Some mammals, like sheep or cicadeer, seem to have internal circannual clocks, like a built-in yearly calendar that gets fine-tuned or entrained by the photoperiod.
An internal clock synced by daylight.
Right. But most mammals studied, like various mice species, seem to continuously adjust. They stay reproductive as long as days are long enough and shut down if days get too short for too long. Though they can become unresponsive or refractory if exposed to the same cue for too long.
And how does the body actually read the day length?
A key player is the hormone melatonin. It's secreted by the pineal gland, but only at night. So the duration of melatonin secretion effectively encodes the length of the night and therefore the length of the day. This pattern acts like a signal throughout the body.
We can see that link really clearly with latitude, can't we? Like those paramastis mice you mentioned.
Exactly. Studies show dramatic differences. Mice populations at lower latitudes might have much longer breeding seasons and fit in more generations per year compared to populations of the same species living much further north, where the favorable season is.
It's not just hormones and environment, though. Genetics plays a part in complex ways, too.
Oh, absolutely. Sometimes in really unexpected ways. Take the white-throated sparrow. Researchers found this fascinating thing called a supergene.
Super gene.
Yeah, it's a big chunk of a chromosome, like 1,000 genes. They got inverted somehow during evolution. Because of that inversion, all those genes are inherited together as one giant block.
Okay, so what does this block of genes do?
In these sparrows, it links distinct traits together, things like the color of their head stripes, specific mating behaviors, even how much parental care they provide. They come as a package deal because of this supergene.
Wow. So completely different traits are locked together genetically.
Exactly. Which suggests that sometimes complex suites of traits can evolve together as a set, not just one gene at a time. It really makes you wonder, how often are these complex behaviors and physical traits linked by such intricate genetic mechanisms?
That's mind-bending. So we really have to consider the whole picture: hormones, photoperiod, genetics, even social interactions.
Definitely. You can't understand reproduction in isolation. Think about male elk needing to physically dominate rivals to mate, or dragonflies defending territories. It's all part of the reproductive strategy.
And what about less predictable environments, like deserts?
Right. In places like deserts, animals might rely on more episodic cues, like a significant rainfall event, rather than predictable cycles like photoperiod. And along coasts, you see animals timing things with lunar or tidal cycles, too.
Okay, so we've got the timing down, both internal and external. But let's zoom out again. A really fundamental question in an animal's life is, how many times do they get to reproduce? Once or multiple times?
That's a huge life history decision, evolutionarily speaking. And it leads to two main strategies. First, there's similarity.
Similarity.
These are species physiologically programmed to reproduce only once in their lifetime, and then they typically die.
Just one shot?
One big shot. Think of the nereid worm transforming into a reproductive form, spawning, then dying under hormonal control, or an octopus guarding its massive clutch of eggs, foregoing food, and dying after they hatch.
Incredible dedication.
Or mayflies, living as aquatic nymphs for ages, then emerging as non-feeding adults just to mate and lay eggs in hours or days. And the classic example, Pacific salmon making those epic upstream migrations, breaking down their own body tissues for energy, spawning, and dying.
That salmon journey is just iconic.
It is. And there's even a rare mammal example, the male antechinus, a little marsupial mouse. The males go through this incredibly intense, frantic mating period, and then they just die. Yeah. Often from stress-induced immune system collapse.
Wow. So the bottom line for similarity is maximum investment in that one reproductive event.
Exactly. The parent sacrifices everything, its own future survival and vigor, for that single batch of offspring. All in.
Okay, so what's the alternative?
The alternative, and what most animal species actually do, is heteroparity.
Heteroparity, multiple times.
Right. Physiologically capable of 2 or more distinct reproductive periods in their life. Think birds nesting year after year, mammals having litters annually. For these species, it's a different calculation.
How so?
Well, heteroparous parents have to balance investment in their current offspring with preserving their own health and survival so they can reproduce again in the future. They're playing the long game, thinking about lifetime reproductive success, not just one big event.
A more complex balancing act. Okay, so whether it's one shot or many, how do parents actually get resources to their offspring? Provisioning them.
The most basic and common way is packing yolk into the egg.
Right, the yolk.
In many aquatic species, especially those with external fertilization, you see lots and lots of tiny eggs with very little yolk each. Think sea urchins or codfish, releasing millions of eggs.
Millions? Survival chances must be tiny for each one.
Extremely low initially. They run out of that small yolk supply very quickly and have to start finding food almost immediately. But there's an evolutionary trade-off.
Producing fewer but better equipped offspring.
Exactly. Look at something like a dogfish shark. Similar size to a cod, but it produces maybe fewer than 20 eggs. But each egg is huge, packed with yolk. Those offspring have a much, much higher chance of individual survival.
So quantity versus quality, essentially.
A classic evolutionary trade-off. Many small eggs, low individual chance, few large eggs, high individual chance.
Are there other ways to provision beyond just yolk?
Oh, lots. Prenatally, you have placental mammals, of course, where nutrients transfer directly from the mother's blood. But some lizards and sharks have evolved placenta-like structures too.
Really? Lizards?
Yep. And then there's a strategy called nurse eggs. The mother lays extra eggs that aren't meant to develop. They're just food for the developing siblings. Some snails do this, and maybe the most extreme version, some sharks, like great whites, practice intramaterine cannibalism, where siblings eat each other in utero.
Whoa. Okay, that's intense provisioning. What about afterbirth or hatching?
Postnatal provisioning is huge, too. Obviously, lactation in mammals is key. Birds bringing food back to the nest. Even wasps that paralyze a spider lay an egg on it and seal it in a burrow. That spider is postnatal provisioning for the wasp larva.
It all comes at a cost to the parent, though, right? All that energy.
A massive cost. Provisioning helps the offspring immensely, but it drains the parents. Studies on small mammals like mice show that lactation is often the single most energetically expensive period for the mother, more than doubling her energy needs. It's a huge investment. If we connect this to the bigger picture, it's a testament to the immense energy investment animals make to ensure the survival of their young.
Okay, so the eggs are provisioned, the offspring are ready. How does fertilization itself happen? We mentioned external versus internal earlier.
Right. External fertilization is generally considered the more ancient state. Common in aquatic animals, especially those releasing lots of small eggs, sperm and eggs are just shed into the water, and fertilization happens out there.
Simple, but maybe risky.
Can be. Dilution, currents, predators. Internal fertilization has evolved independently many times, and it's essential for a couple of key developments.
Such as?
1. Producing shelled eggs, like in birds, reptiles, insects. Fertilization has to happen internally before the shell gets added. 2. Internal development viviparity, like in mammals, where the embryo develops inside the mother.
And how do they manage internal fertilization? Different methods.
Lots of ways. The mammalian penis is one way to deliver sperm. Birds often use a cloacal kiss, just pressing their openings together. Many invertebrates, and sharks too, use spermatophores, packaged bundles of sperm that are transferred to the female.
And once the sperm are inside, it's still a long journey for them.
Oh yeah. Millions might enter, but relatively few reach the vicinity of the egg. And even then, they're not quite ready. Mammalian sperm typically need to undergo capacitation within the female reproductive tract.
Capacitation.
It's a final maturation process, like getting them fully activated, enhancing their swimming ability, and preparing them to fuse with the egg membrane.
And if multiple males have mated with the female?
Then you get sperm competition. It's
Yeah.
literally a race between sperm from different males to fertilize the egg. This evolutionary pressure has led to things like males in some species developing dramatically larger tests relative to their body size just to produce more sperm and increase their odds.
A literal arms race at the sperm level. Okay, we've talked a lot about timing. How flexible can animals be? Can they decouple different parts of the process?
They can be remarkably flexible. Decoupling steps is a common strategy to sync reproduction with the best environmental conditions. One major way is sperm storage.
Females storing sperm.
Exactly. The female mates, but stores the sperm, sometimes for incredibly long periods, only using it to fertilize her eggs later when conditions are optimal.
How long are we talking?
It varies hugely. Female blue crabs can store viable sperm for over a year after just one mating encounter. Queen honeybees, for years. Some bats that mate in the fall store sperm all winter to fertilize eggs in the spring. That's over half a year.
Wow. Here's where it gets really interesting. Imagine having that kind of long-term storage. That's incredible flexibility.
It absolutely is. Another key decoupling mechanism is embryonic diapause, often called delayed implantation in mammals.
Diapause, like a pause in development.
Precisely. It's a programmed state where the embryo temporarily stops or dramatically slows its development. This creates a gap between fertilization and the actual completion of gestation. It's evolved independently many times.
In insects, too.
Oh, yes. Classic examples are silkworm moths or some locusts. They lay eggs in autumn that are programmed to enter diapause. They often need a prolonged period of cold exposure to break diapause, ensuring they only hatch in the spring when food will be available.
Clever. And in mammals.
In mammals, it typically involves the early embryo, the blastocyst, pausing its development before implanting in the uterine wall. This delayed implantation can be obligate or facultative.
Obligate meaning it
Yes.
always happens.
Yes. Part of every normal pregnancy for that species. Antarctic fur seals are a great example. They mate shortly after giving birth in early summer. But implantation is delayed for about 3-4 months. Their actual placental development takes about 250 days. That delay ensures the pup is born exactly 1 year later, back in the favorable early summer conditions.
Perfectly timed and facultative.
Means it can happen, but doesn't have to, depending on the circumstances. White-footed mice do this. If a female gets pregnant again while she's still nursing a previous litter, which often happens because many rodents have a postpartum estrus.
Meaning they can mate right after giving birth. Rabbits do that too, right?
Exactly. Rabbits, hares, seals, many rodents. So if that mouse conceives during postpartum estrus while still nursing, the implantation of the new embryos might be delayed. This staggers the development, preventing her from having to meet the huge energy demands of late pregnancy and peak lactation simultaneously. It spaces out the litters.
That makes so much sense managing energy resources, so we see all sorts of annual timing strategies.
We do. Besides seasonal breeders, you have monostrous animals that only have one reproductive cycle per year, like those Pololo worms with their amazing synchronized spawning link to the moon, or red foxes.
Just one chance a year.
Right. And then large mammals with long gestations have unique challenges. Sheep are short day breeders. They become fertile as days shorten in autumn, ensuring their 5-month gestation leads to spring births. Zebras and horses have roughly 12-month gestations, so they tend to breed such that birth aligns with favorable seasons year after year.
And the really big ones.
Elephants are extreme 22 months gestation, plus nursing for maybe 3 years. They might only give birth every 6 or 7 years. Large whales are similar, maybe 1.5-year gestation, birth every 2-3 years. It's all about matching these long cycles to resource availability.
Okay, this is incredible. Let's dive deeper now into the hormones, especially in mammals where we know quite a bit, right? Starting with the female side.
Right. Female mammals operate on cycles. We talked about the ovarian cycle. Follicles develop, follicular phase. An egg is released, ovulation. And then the structure left behind, the corpus luteum, takes over, luteal phase.
And this links to menstrual versus estrous cycles.
Exactly. They reflect the same underlying ovarian events, but the outward signs differ. In estrous cycles, the key outward sign is estrus, or heat, that period of sexual receptivity which is tightly synchronized with ovulation. If pregnancy doesn't occur, the uterine lining is typically resorbed. In menstrual cycles, like in humans and other primates, menstruation, the shedding of the uterine lining happens long after ovulation, only if fertilization fails. Receptivity isn't as tightly linked to ovulation.
Okay, so inside the ovary, during that follicular phase, what's happening?
You start with tiny primordial follicles. Under hormonal influence, some start growing, becoming primary, then secondary follicles. Usually, in humans at least, only one becomes a fully mature, dominant Graafian follicle destined for ovulation. The others undergo atresia, basically degeneration.
And hormones are driving all this.
Absolutely. It's the hypothalamus-pituitary-ovary, HPO axis. The hypothalamus releases GnRH impulses. Those pulses tell the anterior pituitary to release LH and FSH.
LH and FSH, the gonadotropins.
That's them. They travel to the ovaries. LH primarily acts on cells called the tecocells in the follicle, stimulating them to produce androgens, testosterone-like hormones.
Okay.
FSH acts mainly on the granulosa cells surrounding the oocyte. And here's a key interaction. The granulosa cells, stimulated by FSH, take the androgens produced by the tecocells and convert them into estrogen using an enzyme called aromatase. It's called the two-cell, two-gonadotropin model.
So the follicle cells cooperate to make estrogen.
You do. Estrogen levels build slowly at first, then rapidly increase as the dominant follicle grows. Estrogen feeds back to stimulate more granulosa cell growth. Initially, low estrogen tells the pituitary to ease off LH-FSH, slightly negative feedback.
But then it switches.
Yes. Critically, when estrogen levels get very high for a sustained period, the feedback switches to positive. High estrogen stimulates specialized neurons in the hypothalamus-Kisspeptin neurons, which then drive a massive release of GnRH, leading to that big LH surge and a smaller FSH surge.
Ah, the Kisspeptin neurons are the switch.
They seem to be a key part of that switch mechanism. The granulosa cells also produce inhibin, which specifically suppresses FSH release, helping ensure only one follicle usually dominates.
And while the ovary is doing this, the uterus is preparing too.
Yes, perfectly coordinated. The uterine lining, the endometrium, goes through phases. Early on, driven by rising estrogen, it's the proliferative phase. It thickens, gets more blood vessels. After ovulation, under the influence of progesterone from the corpus luteum, it enters the secretory phase. Glands start secreting nutrients. It becomes receptive for implantation.
So the LH surge causes ovulation itself.
Correct. The LH surge triggers final oocyte maturation, stimulates progesterone production even before ovulation, causes the release of enzymes to break down the follicle wall, and ultimately leads to the rupture of the follicle and release of the egg.
And the leftover follicle becomes
The corpus luteum. This temporary endocrine gland is vital. It pumps out progesterone, the dominant hormone now, plus estrogen and inhibin. Progesterone maintains that secretory state of the uterus, inhibits uterine contractions, basically keeps everything ready for pregnancy.
And if pregnancy doesn't happen?
If no embryo implants and signals its presence, the corpus luteum degenerates after about 10-14 days in humans. Progesterone and estrogen levels plummet, the negative feedback on the hypothalamus pituitary is removed, FSH starts to rise again, and a new cycle begins.
Such an intricate, self-regulating loop. Okay, let's switch gears to the male side. It seems simpler, maybe. Continuous production.
In adults during the breeding season, yes, sperm production, spermatogenesis, is typically continuous, unlike the female cycles. The tests are the site, of course.
And they're usually in the scrotum, outside the main body.
Right, and that's crucial. They need to be a few degrees cooler than core body temperature for viable sperm production. Why exactly sperm need that lower temperature is still a bit of a mystery, but they do. Marine mammals with internal tests have evolved amazing countercurrent heat exchangers in their blood vessels to keep the testes cool.
Wow. Okay, inside the testes.
Sperm are produced in long coiled tubes called seminiferous tubules. These make up most of the testes volume. Within these tubules are the developing sperm cells and large Sertoli cells.
Sertoli cells. What do they do?
They're essential support cells. They nourish the developing sperm, regulate the process, form a barrier, secrete fluid, produce inhibin, which suppresses FSH, just like the females, and also make androgen-binding protein, ABP, which helps keep testosterone levels really high right there in the tubules where it's needed. They're influenced by both FSH and testosterone.
And where does the testosterone come from?
From the Leydig cells, which are located in the connective tissue between the seminiferous tubules.
And the hormonal control loop. Same hormones from the brain.
Same top-level hormones. GnRH from the hypothalamus, LH and FSH from the pituitary, but they have different targets. LH specifically targets the Leydig cells, stimulating them to produce and secrete testosterone.
Okay, LH makes testosterone, and FSH.
FSH primarily targets the Sertoli cells, supporting their function and aiding spermatogenesis, though testosterone itself is the absolutely critical hormone needed for sperm production to occur properly.
And testosterone has broader effects, too.
Oh, yes. It drives masculinization during development and puberty growth of penis and testes, voice deepening, facial hair, increased muscle mass. And just like in females, testosterone exerts negative feedback on the hypothalamus and pituitary, keeping GnRH, LH, and FSH levels relatively stable in adult males, which maintains that continuous sperm production.
And the final steps, erection and ejaculation.
Right. Erection is a vascular event. Parasympathetic nerves release nitric oxide and NO in the penis. NO causes smooth muscles in the walls of arteries to relax, allowing blood to flow in rapidly and engorge the erectile tissues.
Nitric oxide. That's what those ED drugs target.
Exactly. There's even a positive feedback loop where the initial blood flow stimulates the endothelial cells lining the blood vessels to produce even more NO. Drugs like Viagra work by inhibiting an enzyme that normally breaks down a downstream messenger molecule, cyclic GMP, allowing the effects of NO to last longer and be stronger.
Fascinating. And ejaculation.
Ejaculation involves the expulsion of semen, which is a mix of sperm from the testes via the epididymis and vas deferens, and fluids from accessory glands, the seminal vesicles, prostate gland, and bulbourethral glands. These fluids aren't just filler. They provide fructose for energy, buffers to counteract vaginal acidity, clotting factors, enzymes, and maybe even signaling molecules.
Okay, so putting it all together, fertilization, pregnancy, birth, the grand finale.
It really is. Fertilization typically happens up in the oviduct or fallopian tube. Sperm arrive, hopefully capacitated and ready.
Capacitated, right.
When one sperm successfully fuses with the egg membrane, it triggers the cortical reaction. Granules just inside the egg membrane release their contents, altering the egg's outer layers to prevent any other sperm from getting in the block to polyspermy.
Only one winner.
Only one. The fertilized egg, now a zygote, starts dividing as it travels down the oviduct towards the uterus, becoming a blastocyst.
And then it needs to implant.
Yes. The blastocyst hatches from its protective outer layer, the zona pellucida, and then burrows into the receptive uterine endometrium. Specialized cells on the outside of the blastocyst, the trophoblast, secrete enzymes to help with this invasion.
But the cycle wants to end. The corpus luteum wants to degenerate. How is pregnancy maintained?
Luteal rescue. The corpus luteum must keep making progesterone for early pregnancy. How this happens varies. In primates, including humans, the very early embryo's trophoblast cells start secreting a hormone called chorionic gonadotropin, CG, specifically human CG, or HCG.
HCG. That's the pregnancy test hormone.
That's the one. HCG acts like LH, signaling the corpus luteum to stick around and keep making progesterone. In rodents, it's a bit different. The pituitary hormone prolactin plays a key role in sustaining the corpus luteum early on, triggered by mating.
Okay, so the corpus luteum is safe. What about the placenta?
The placenta develops as pregnancy progresses. It's an amazing composite organ formed from both embryonic tissues derived from the trophoblast and maternal uterine tissues, the endometrium.
And its main job is exchange.
A crucial job is facilitating exchange between mother and fetus. Oxygen, nutrients, antibodies go to the fetus. CO2 and waste products go back to the mother. Their bloodstreams come into very close contact for efficient diffusion, but they don't normally mix directly.
And it makes hormones, too.
Yes, the placenta becomes a major endocrine organ itself.
Yeah.
Takes over progesterone production from the corpus luteum later in pregnancy, makes lots of estrogen, and produces hormones like placental lactogen, which helps prepare the mother's body and mammary glands for lactation.
Okay, fast forward. Birth, parturition. How does that get started?
Towards the end of pregnancy, there are preparatory changes. Rising estrogen levels stimulate the uterine muscle, myometrium, to synthesize proteins called connexins, which form gap junctions between muscle cells. This allows them to contract in a coordinated way. Estrogen also increases the number of oxytocin receptors on those muscle cells, making them more sensitive.
So the uterus gets ready to contract powerfully.
Exactly. In some mammals, another hormone, relaxin, helps soften the cervix and ligaments in the pelvis to allow easier passage of the fetus.
And the trigger for labor itself.
It's complex, likely initiated by signals from the maturing fetus, but the main drivers of contractions are the hormone oxytocin, released from the mother's posterior pituitary, and prostaglandins, produced locally in the uterus. These cause the powerful rhythmic uterine contractions.
And it builds on itself, right? Positive feedback.
Classic positive feedback loop. Contractions push the fetus against the cervix. Stretch receptors in the cervix send signals to the brain. Brain releases more oxytocin. Oxytocin causes stronger contractions, more pressure on the cervix, and so on, escalating until the baby is delivered. Then the placenta, the afterbirth, is delivered too, and estrogen and progesterone levels drop dramatically.
And finally, lactation. The defining mammalian trait.
Indeed. And often, as we said, the most energetically costly phase for the mother. It involves two main processes: milk production, or synthesis, by epithelial cells lining tiny sacs called alveoli in the mammary gland, and milk ejection, or letdown, where the milk is actually expelled from the alveoli into the ducts.
And that first milk is special. Colostrum.
Yes, colostrum is produced for the first few days. It's lower in fat and sugar than mature milk, but incredibly rich in antibodies, proteins, and vitamins, providing crucial passive immunity to the newborn.
Hormones control this too.
Absolutely. Milk secretion is primarily stimulated by prolactin, released from the anterior pituitary. Prolactin release itself is inhibited by dopamine from the hypothalamus, but stimuli like suckling reduce dopamine release, allowing prolactin to rise.
Okay, prolactin for making milk. What about getting it out?
That's oxytocin again, from the posterior pituitary. Oxytocin causes specialized myoepithelial cells, like tiny muscles wrapped around the alveoli, to contract, squeezing the milk out into the ducts, the milk ejection or letdown reflex.
And suckling triggers both.
Yes, suckling is a powerful neuroendocrine reflex stimulus for both prolactin release to keep milk production going and oxytocin release for milk injection.
And lactation can affect future cycles.
It often does. High levels of prolactin during intensive nursing tend to suppress GnRH release from the hypothalamus. This leads to lactational anestrus, or anovulation basically, preventing estrous cycles or ovulation while the mother is lactating heavily. This is a natural birth spacing mechanism in many mammals, including humans, though its effectiveness varies.
What an absolutely incredible, intricate system. From the boxing hairs right down to the hormonal signals controlling milk letdown, it's just layers upon layers of complexity, all geared towards one goal.
It really is. The diversity of strategies, the precision of the timing, the interplay between hormones, environment, genetics. It's a constant reminder of how finely tuned these life processes are, always shaped by the relentless pressure of natural selection to ensure survival and continuation of the species.
Absolutely mind-blowing.
And thinking about all this intricate synchronization, especially the reliance on environmental cues like photoperiod or temperature, it really makes you pause. It raises an important question for us. In a world changing as rapidly as ours, with climate shifts and habitat alterations, how will animals' finely tuned reproductive strategies manage to adapt? Their very survival is tied to getting that timing right.
That's a really crucial and somewhat worrying thought to end on. The connection between this deep physiology and the challenges of conservation is stark.
It truly is.
Well, this has been an absolutely fascinating dive into the world of animal reproduction. Huge thanks for guiding us through that complexity. And thank you, our listener, for joining us today. We really appreciate you being part of the Last Minute Lecture family. We hope this sparks your curiosity even further. Maybe the next time you see animals in the spring, you'll see more than just behavior. You'll glimpse incredible biology driving it all. Until next time, keep that curiosity alive.